One of the longest-running questions about this pandemic is a simple one: where did it come from? How did a virus that had seemingly never infected a human before make a sudden appearance in our species, equipped with what it needed to sweep from China through the globe in a matter of months? Analysis of the virus's genome was ambiguous. Some analyses placed its origin within the local bat population. Others highlighted similarities to pangolins, which might have been brought to the area by the wildlife trade. Less evidence-based ideas included an escape from a research lab or a misplaced bioweapon. Now, a US-based research team has done a detailed analysis of a large collection of viral genomes, and it finds that evolution pieced together the virus from multiple parts, most from bats, but with a key contribution from pangolins. In cells, recombination is a normal part of genetics. Anytime two DNA molecules share extensive similarities, it's possible for them to exchange pieces. The result is a hybrid molecule, a stretch of DNA from one parental piece of DNA, followed by a stretch from the other. As a result, some of the differences between the two parent molecules get scrambled, some from each parent will end up on the final molecule. Recombination is a normal part of the reproduction of complicated cells. If you happen to have an offspring, you've given that child a set of chromosomes that are a mix of pieces from the ones you were given by your mother and father. Recombination can also take place in simpler cells, where it has been the primary tool that we've used to engineer new or altered genes into the genome of bacteria. And, since the molecules that perform the recombination aren't especially picky about which DNA molecules they work with, DNA viruses that infect cells can sometimes recombine if more than one strain of virus infects a single cell. The answer is it. But other processes basically perform the same function: mix RNA pieces up to form different genetic combinations. For example, influenza virus difference of its genome on 8 different molecules, allowing cells to be infected more than a flu virus strain to create viral particles with a random molecule from 2 strains. Basic genome analysis has confirmed that SARS-CoV-2 is most relevant to some of the separated viruses from bats. But different areas of viruses are more or less related to different bat viruses. In other words, you will see a long RNA strip similar to a virus from a bat, but then it will suddenly convert to look the most with a different bat virus. A little other information to get rid of this study is a sign of where the changes in the protein of the virus can be used. This does not have the ability to endure changes in a genome area, tends to be a sign that proteins encoded by the genome part has an essential function. Researchers have identified some of these, one of which is part of the spike protein derived from pangolin virus. In all 6,400 SARS-CoV-2 genes isolated in a pandemic, only 8 from a case cluster have any changes in this area. Therefore, finding pangolin strains is essential for the ability to target viruses. All that shows that the extra pandemic is a question of time, instead of if. But, of course, has been proposed in the consequences of the original MERS and SARS, and the entire world had a little significant to study risks, work for treatment or pandemic plans come. We can only hope that the clearer example of COVID-19 will change that.